Welcome. Today is chapter 5, structure and function of plasma membranes. Some of this will be reviewed from lab and some of this will be adding to that. So we'll talk about components and structure of the membrane. We'll talk about the difference between passive and active transport. We'll mention some proteins that are used for the transport of materials. And then finally, when the cell needs to transport in things too large for these proteins, then they'll use bulk transport, and we'll look at that. Okay, so study tip reminder about scheduling enough time regularly, hopefully daily, maybe every other day, but you definitely need to put in time regularly. Do not wait for the week before the exam to do your studying. So are you studying every day? Are you studying enough time and enough different strategies? So you don't want to entirely skip over stuff that seems easy, but you certainly do want to give more time, attention, energy to new or challenging concepts. And make sure that you're actually giving yourself a true test if you remember and understand content. So things like writing out your answers or typing out your answers to questions before you look at the answer is a good way to see, so you'll actually see where you're at. And just because you remember something, if it's just a memory test, just because you remember it one day doesn't mean you'll remember it next week. So make sure that you're constantly going back and reviewing and checking your memory on your terms, for example, terminology. For understanding, you have questions that you answer for homework. Are you able to answer those without looking up at any of your notes or your book? Right, that's a good way to check your understanding. Okay, plasma membrane functions. As you know, the plasma membrane, every single cell has a plasma membrane. It defines the outer border of cells, and if it's eukaryote, which we'll be focusing on today, just eukaryotes, then the organelles are also lined with plasma membrane. So the plasma membrane helps determine what's able to enter or exit the cell or the organelle. It also, for the plasma membrane, it receives external signals and can initiate internal or cellular responses. It also can serve as a way for cells to adhere to one another and form tissues. Okay, so as you know, phospholipids are what we call a phosphate head and two fatty acid tails. The phosphate head is attached within the head. The phosphate group is attached to glycerol. And for your fatty acids, we typically draw it as one being saturated and the other one being unsaturated. And those would be the hydrophobic region, right? They do not interact with water. But in reality, each fatty acid could be saturated or unsaturated. So it does, there is some variation within the cell. And as you recall, if they were all saturated, they would be able to pack very tightly together. And whenever they're unsaturated, that's going to make that kink or bend in the tail, and it's going to take up more space. Okay, so think about putting some oil in some water. The oil is going to form all those droplets and end up floating at the top and try to stay away from the water itself. Oil and water doesn't mix. So when you're talking about these phospholipids, you have the head, the phosphate glycerol head is actually hydrophilic, and it can and does interact with water. It's just the tails that are acting like oil, right? Hydrophobic. So when you put phospholipids into water, they're going to, all the tails are going to orient together to hide from the water, and all the heads are going to be on the outside facing the water. And so we call this a micelle. It's like a droplet. It's this specific arrangement where all the tails, the hydrophobic tails, are staying together away from the water. Okay, so this is energetically favorable. This happens naturally. Okay, and only when in an aqueous or water solution, okay, water-based solution. The polar heads, the phosphate, oh, excuse me, lots of yawns today. Okay, polar heads, the phosphate group and glycerol group, those are hydrophilic, so they're going to be facing outward towards the water. The tails, the fatty acid tails, are hydrophobic. They're going to stay inside away from the water. Now, think about cells. Cells are doing the same thing, with the difference being that the inside of the cell is also aqueous. So that's why we have a second layer, and it's the lipid bilayer for a cell membrane, right? So the second layer allows the heads to face the inside aqueous solution and the heads to face the outside wet environment. We are going to watch this video together. All the other video links that I've provided today, I will trust you to watch them on your own. You can even pause the video to watch them as you go through the lecture, which is nice. Okay, so let's go ahead and watch this.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Okay, you can rewatch that again. I've provided the link for you right here in the slides. I suggest you do watch it again because though short and seemingly simple, it really hits on everything that our lecture is hitting on today. Okay, so as it mentioned, the fluid mosaic model, this is the ability of most of the proteins embedded in the membrane are able to move around. So you see these, here's a protein with a sugar attached to it called the glycoprotein. And then here's another protein here, right? So all the proteins in this image are shown in blue. Some of them are on the surface, some of them span. So all these different types of proteins and other molecules as well that are embedded into the membrane typically can move around. And the lipids themselves are also pretty flexible, as you saw in the video. So this is the idea of lots of different components mixed together form the mosaic, and then the fluid part, because it is flexible, or things are able to move. They're not stuck in place. They're not rigid and stationary, but they're fluid, flexible, movable. Okay. So that's what we mean by the membrane fluidity. Okay. We can increase the fluidity and we can decrease the fluidity by certain conditions. We can also think of membrane fluidity as the ability for substances to pass through the membrane, but that really would be permeability. A lot of the factors we're going to talk about that increase fluidity also will increase permeability. So try to keep those two terms straight. They are different terms. So fluidity, the ability of the substances in the membrane to move around, whereas permeability is the ability of a substance to pass through the membrane and get to the other side. Okay, so the membrane needs to be flexible, as you saw in the video where they poked it and it kind of moved, it didn't break apart, right? The little bit that seemed to break apart reformed very quickly. So it needs to be flexible, but not so flexible that it completely loses its structure, right? Okay, so fluidity can be affected by the type of phospholipid, like thinking about those fatty acid tails, temperature, how hot or cold is it, and how much cholesterol have you embedded. So let's go ahead and think about each of these three. So the phospholipid type, again, is referring to the type of fatty acid. So if you have all saturated fatty acids, they're going to pack together very closely, very tightly, and it's going to be more rigid. If you have unsaturated fatty acids, they will have kinks and bends, right? And they will take up more space, and then they would be less close, they would be spread apart, and that's going to make it more fluid. So we typically have a combination. You don't typically have all of the phospholipids one type, right? You'll have a combination to adjust for an intermediate amount of fluidity. Temperature, when it's really cold, makes things slow down and can, so just like it decreases the kinetic energy and causes molecules to move more slowly, it can also compress the molecules of the membrane, making them more rigid. But by adding cholesterol, we think of cholesterol as like a buffer. For membranes, it's a fluidity buffer. And the cholesterol would be embedded in the fatty acid layer, so with the fatty acid tails. And it actually helps it not to get too rigid when it's cold, but also not to get too fluid when it's hot. Of course, this has its limit, just like pH buffers can only do their job to a certain point. Eventually they'll give way, but it does this is why we're able to tolerate such a large range in temperature, whereas things like bacteria, they have a much more narrow range of temperature that they can tolerate. Okay, so membrane proteins. In this picture, we have a protein where it goes from the inside of the cell to the outside of the cell. It passes all the way through the membrane a single time. So this is called a single pass. Proteins that span the at least one whole layer of the bilayer are called integral proteins. So this is a single pass integral protein. It actually is also considered a trans membrane because it goes through both layers. It goes from one end all the way to the other side. But you can be an integral protein like this right here. This monotropic protein is still an integral protein. It just is not transmembrane because it doesn't go through to the other side. It only goes through one of the layers. Okay. Then you have this extrinsic protein, which is not an integral protein. It's only on the top. We're going to see that next. We're going to call those peripheral membranes. Okay, so the integral proteins integrate completely into the bilayer. This is a little bit of a confusing definition. This is what your book uses. But I would like you to make a note that that means it only has to go through one of the layers of the bilayer. So the monotropic protein is still considered an integral protein, whereas something that's only on the top or only on the bottom would be a peripheral protein, only on the surface. Okay, so integral proteins, I talked to you about the single pass. In this picture, we have something called a multipass. There's lots of units of the protein that embed in the fatty acids. And so that would be the multipass. And you could have something that's double, or in this example is triple. Anything more than 3 passes, we would just call a multipass. So we usually only say single, double, triple, and then after that, it goes to multi. Okay. So as I mentioned, some integral proteins span only a single lipid layer. It's not shown here, but on the previous slide, it is shown right here. So this would be that single, spanning the single layer of the lipid bilayer. Transmembrane proteins are shown here. All of these are transmembranes because they go from one side all the way to the other side. So they have a inside and an outside region, inside the cell, outside the cell, the yellow part being the membrane. Okay, integral proteins are typically considered permanent parts of the membrane, meaning even though they might move around a little to the right or the left, or forward, backwards, right? They might move around within the membrane, but they'll always be in the membrane. Whereas peripheral proteins can be added and removed pretty quickly. These are typically always going to be there. All right, so that is integral proteins. Peripheral proteins, as I mentioned, will be only on the surface. That might be the outside of the cell, or it could be inside, but it won't be both. It'll be one or the other. And sometimes it looks like it's going into the fatty acid tails, but this is actually an additional component to anchor it into the membrane, where the protein itself is still only on the surface. But that's okay. We're not going to worry about getting too detailed. We just want to know peripheral proteins occur only on the surface. They're either attached to integral proteins or they're attached directly to the phospholipid heads. Okay. They are considered temporary, which makes them easy for the cell to attach, detach, and reattach at specific times under specific conditions to carry out specific cells for specific functions. So this gives the cell a lot of control to regulate itself. Okay, so this allows cells to coordinate and communicate with a network of proteins and reactions. We'll see some examples in future lectures. Okay. One important example of a peripheral protein that I wanted to bring to your attention that I was a little surprised that your book didn't include is called major histocompatibility complex. If you've ever heard of people that need to have an organ transplant and they're on a list waiting for a match, a donor to match, this is primarily what they're talking about. There are additional factors, but the very first thing that they must do is match this surface protein, peripheral protein on the cells called MHC, major histocompatibility complex. This name, major histocompatibility complex, is from this actual scenario of transplants where they found when they transplanted tissue into another, like from one mouse to another mouse, for example, that it was rejected, that there was a major immune response. The immune response attacked the foreign tissue and killed it. And usually that would also lead to killing the mouse. And so this is how we know, well, this and actual experiments on people, unfortunately, is how we knew that there's a factor that allows cells to recognize self. And if that factor is not present, the immune system attacks. And so we call this factor the MHC. Okay. After further studying, we actually were able to identify the specific MHC molecules. And now we know that there's two types, type that is on all of the normal cells in the body. We say nucleated cells because red blood cells are not nucleated and don't have MHC. Everything else will have MHC class 1 and additional MHC class 2 on specialized immune cells called antigen presenting cells. These are things like macrophages, for example, and B cells that are involved in specific processes of the immune system. So they would have both class 1 and class 2. Everything else would just have class 1. And this will come up time and time again as you take biology classes. So it is important to take the time to learn this. But also if you ever have to deal with yourself or a loved one needing a transplant, it can be helpful for you to understand this. Okay, blood is not nucleated blood cells. Red blood cells are not nucleated cells. They don't have MHC, but they do have a different type of receptor, different type of peripheral protein, which you know is like the ABO plus or not plus. So you can be O negative or O positive, and you could be right type A positive or negative, right? So all of those are different proteins found on red blood cells. So whenever talking about donating blood or receiving blood, you would make sure that you're a match for all of those proteins for the blood, right? Just like a tissue donation needs to be a match for MHC. Okay. So some of our proteins will have carbohydrates added onto them, and sometimes carbohydrates will be embedded directly into the membrane. And so the third major component of membranes, so we talked about the phospholipids being the primary, proteins are the second, and then carbohydrates would be the third major component. And we sometimes we'll call these oligosaccharides because they're small, oligo for small or short, saccharide for sugar, right? So oligosaccharides, these will be on the exterior surface only. So please don't confuse this with peripheral proteins. So peripheral proteins can be on the top or the bottom, right? Outside or inside on either surface. But the carbohydrates will only be on the exterior surface. This is to help either bind proteins and form glycoproteins. They could be bound to lipids, forming glycolipids. Their purpose, collectively, all of the carbohydrates on the surface are called the glycocalyx, that sugary coat we talked about before. This is going to do things like attract water to the cell surface. This is very beneficial for the cell. The specific proteins can help in addition to the MHC proteins that we saw. The carbohydrates can also be a form of identification, and they can also help, these carbohydrates can also help cells to attach to one another to form tissues. So lots of really important functions. So they create the extracellular environment, or matrix. Plasma membranes are called asymmetric because the inside surface does not match the outside surface. They're very different. Okay. Interior proteins can anchor fibers of the cytoskeleton to the membrane, where exterior proteins bind to the extracellular matrix. Glycoproteins bind substances that the cell needs to import. So you see here on this example, you have a glycolipid here, you have, right, a glycoprotein here, and this, any of these extensions away from the surface can bind particular molecules, and that can serve as either a signal, or it might then lead to taking in a substance. Membranes can be damaged. We've talked about this before. Alcohols, we talked about how alcohol can disrupt a membrane. Detergents, that's why washing your hands with soap and water helps to kill germs. You get the physical removal, but you actually will kill some as well. Antibiotics, if you take antibiotics, certain antibiotics will target the membrane and damage it. So all of these alcohol, detergent, and certain antibiotics, when they damage the membrane, they typically don't remove the entire membrane, but they cause little weak areas that lead to leakage of the cell. So the cell contents will leak out. If there's enough of it, if you cause enough weak spots where you create enough osmotic pressure, you could lead to the complete bursting of the cell. But typically it would just be the leakage. And as the cell loses its contents, it will, unless it's very short, very tiny, it typically is going to cause the cell to die. Okay, so hand sanitizer works on this principle. Hand sanitizer is made of rubbing alcohol or isopropyl alcohol. But any alcohol that is concentrated enough will work. So lab grade ethanol tends to be 70%. It's not purified. It has lots of toxic things in it. So people have, joked about drinking the lab ethanol to get drunk, and it will get you drunk, but it can also leave you blind. It can make you very, very sick and other problems because of those toxins. So never ever drink lab ethanol. As far as alcohol that you do drink, not sure that it would be strong enough to disinfect anything. because it would need to be 130 proof or more. Yeah, that's a lot stronger than what you usually have, I think. Alcohol requires some water to be effective. So though you do want it to be strong, you want that the target is 70%, which would be, I think 140 proof, but technically 65, anything stronger than 65% will work. If you don't have enough water, like say you go to just pure alcohol, 100%, it's going to evaporate very, very quickly. And it's not going to interact with and insert itself into the membrane, and it's not going to be able to cause that leakage. So you do need some water, so you can use anything from 65 to 95%. Okay, transport. So the plasma membrane, we learned already that it has a fluid mosaic model, being flexible, fluid, right? Now we're thinking about transport, getting things from one side to the other. So we say that it's selectively permeable, which means that certain molecules are allowed to pass through and not others. This feature allows the cytosol solutions to differ between the outside extracellular fluid is different from the concentrations that you find inside the cytoplasm. So cytoplasm is all of the stuff inside the cell, whereas the cytosol is just the fluid portion. Transport can be passive. For certain molecules, it's passive, and for other molecules, it is active. If it's active, it requires ATP. If it's passive, it does not require ATP. So we'll first look at simple direct diffusion. We'll look at facilitated diffusion. Then we'll look at active, okay? So the simplest type of passive transport would be directly diffusing across the membrane. This is when substances move down their concentration gradient from high concentration to low concentration. So the net movement from high to low will cease once an equilibrium is reached. So that's what we see here. So here you have a very high concentration on one side and nothing on the other side. So this is as steep of a gradient as it can get. This is going to force these molecules to want to pass through the membrane. And if the membrane is permeable to that molecule, which in this instance it is, then it's going to start to diffuse through. And it's going to continue to have this net movement from top to bottom until an equilibrium is reached. And then we say there's no net movement. You'll occasionally get one or two that pass through, and then another one or two are going to pass back through. So you can continue to have movement, but the amount that goes out will equal the amount that comes back in. So it's no net movement. Examples, what is able to go directly through the plasma membrane? These have to be small and also non-polar molecules. Small, non-polar. So things like oxygen can just go right through. Carbon dioxide can go right through. That makes sense because we were talking about today breathing in air, and that oxygen automatically diffuses through the tissue of the lungs into the tissue of the cardiovascular system, right? Okay. And then also there's the lipid hormones that we looked at, things like testosterone and cortisol, stress hormone. Those can also just go right through because they're lipids. Okay. So what if you wanted to change the rate at which something diffuses. Well, you could just add more, assuming that you're trying to get it from the outside of the cell to the inside. You can make a bigger concentration gradient. The greater the difference, the faster the diffusion, right? So here you're going to get a high diffusion rate. They're going to start moving across very quickly. But as you get closer to equilibrium, it's going to slow down. The molecules themselves, if they're small, they're going to diffuse more quickly than if they're big. So you can think of this as you run around a track with and without a backpack, heavy backpack. When you run with your heavy backpack, you're going to run slower. Okay, so smaller molecules diffuse more quickly. Temperature, you heat something up, it has more kinetic energy, it vibrates more, it will move more quickly. So that's going to speed up diffusion. Solvent density, if it's really dense, it's going to slow the movement down. So it's going to reduce diffusion rate. So you can think of this as, swimming in water, you'll be able to move more quickly than if you were swimming in something like, I don't know, pudding. Pudding is thick, it's going to slow you down. Okay, weird example, but it works. Solubility, non-polar, or in other words, lipid soluble substances are going to easily go through the membrane as long as they're small enough. Okay, so if it's soluble, if it's lipid soluble, it will diffuse quickly. Surface area. Oh, excuse me. Increased surface area of the cell or the organelle, if you're talking about going into an organelle, that's going to help speed up diffusion rate. If there's more membrane, then the solute has more space to diffuse through, and it'll speed up the diffusion rate. So trying to diffuse into a tiny organelle is going to take more time than diffusing into the cell as a whole, big cell, right? Okay, distance traveled, farther it has to go, the longer it's going to take, just like in a car, right? Pressure, if you increase the pressure, it will drive or force materials through the membrane faster. So that will speed up. If you reduce pressure, then it's going to take longer. Okay, so what if you are not one of those molecules that can go directly through the membrane? So what if you're an ion, or you're small, but you're polar? Then you're going to need to slide through a protein. This can be a protein channel, or it could be a carrier protein. And we'll talk about each of those in a moment. So this is called facilitated because you're using a protein, but it is still diffusion because you do not need to add energy. You're simply going down the concentration gradient. So it is still diffusion, okay? So channel proteins, we'll see some of them are open all the time, and some of them open in response to that solute being present. So for example, aquaporons are specific to water. They're typically always open, and water can just pass freely through. Whereas things like muscle cells will have specific ions that trigger, we call it a gated ion channel. And when that ion's present, it'll cause the channel to open up, and that will lead to muscle contraction. All carrier proteins are specific. Okay, so not all channels are specific. The examples here were specific, but we do have some channels that are not specific. If it's small enough, it'll just fit through. Carriers, on the other hand, are always specific to just one type of substance. So for instance, you have glucose, transport proteins, they only transport glucose. So if that substance binds to it, it will cause a change in the shape of the protein. It'll push it through to the other side. Many allow movement in either direction. So it's responding to concentration gradient. Some are one direction only. As long as the specific solute's present, it will transport it. So it's specific to only one substrate. It's going down a concentration gradient, so it is still diffusion. Now, osmosis is when we're talking about the movement of water through the membrane. Typically, this happens because the solute is too big to pass through the membrane. And so the only way to make an equilibrium is to have water move. And this is what we did in lab today with the dialysis tubing. The starch was too big to pass through the dialysis tubing. So water actually started to move into the dialysis bag. But your glucose was small enough to travel. So your glucose started to leave the bag and go into the water. And then the iodine was in the water and it was not inside the bag. So it was attracted to wanting to move into the bag, right? To move away from high towards low concentration. And the iodine was small enough to fit through the bag. So this osmosis, though, only refers to the water itself moving. So the example of the iodine moving through the membrane is an example of selective permeability, not osmosis. The example of the glucose, same thing. It's not osmosis, it's the glucose is moving. The only osmosis only talks about water. So when you, for example, if you just have starch, if you were to have left that bag of starch in your water, it would have continued to have more and more water fill up that bag. So that's what we're seeing here in this example. You start out with a membrane here. You have a little bit of solute on one side and a lot of solute on the other. Pretend this is starch. It's too big to pass through the membrane. So instead, water will diffuse, and this takes time. You know, it could take an hour or so, but eventually you'll see the height on this side increase because the water has left. It's gone from the left to the right side. Now that you have a bunch of water here, you have diluted down the concentration of solute to reach an equilibrium. Okay, so osmosis is the diffusion of water and only water across the membrane. Why does this happen? Because there's a difference in the concentration of solute, and the solute is not able to pass through the membrane, only water is. Okay, so water will move towards the higher solute when the solute cannot move. Okay, so water moves from an area of higher water to lower water. This is what your book talks about. I want to make sure you're paying attention though, because if you're talking about solute, you would say water moves towards the higher concentration of solute. If you're talking about water, water is moving from an area of high water to an area of low or little water. Okay, so in this picture, we would say this has a lot of water and this has a little bit of water. Yes, the level is the same on both sides, but the amount of solute in this side compared to this side, we have so much more solute on this side that you have less water to fill that same volume. You cannot fit as much water molecules on this side because you have solute taking up the space, which is why the water moves over time. Okay. So another visual, instead of looking at one side and left and right side of a beaker, you could think of it as a bag. So you could put a bag full of starch, and water is going to be attracted because the starch is trapped. And after some time, water has gone into that bag. The bag is now plump. You also have water level in your tube has increased as well. And this is now we say at equilibrium for the amount of water on both sides is equal. Okay, so water will flow into the sac or bag because the solute concentration inside is greater than the solute concentration outside. So we would say that this solution is hypotonic, right? And that's why water is going to rush in. A cell can prevent this from happening in two ways. It could have a rigid cell wall that only allows a little bit of water to flow in, or it could have a membrane that is non-permeable and controls the amount of flow. Okay. Okay. So tonicity, as we talked about, hypertonic, isotonic, and hypotonic, is describing the osmolarity of a fluid, comparing the extracellular fluid to the inside of the cell or the cytosol. Okay, so we say that tonicity is like a reflection of the osmolarity of a solution, and it's that comparison. Okay, and as you saw in lab today, red blood cells burst open if placed in a hypotonic solution because there's so much solute inside the cell that water is going to rush in. And then there's no cell wall, so it will end up bursting open before any equilibrium is reached. If there was enough solute inside the solution to just for a little bit of water to go in, then it wouldn't burst open. Okay, but we looked at extreme hypotonic examples where it did burst open. Isotonic means you have the same amount of solute outside and inside. So the amount of water going in is going to be equal to the amount of water going out. And there's no change, no net flow. So it's a normal, happy red blood cell. If you put it in a hypertonic solution, there's a lot of solute outside the cell, causes water to leave the cell, and that causes the cell to shrivel. And the technical term that you need to know for that is crenate, right? Crenate, it was written on the board in lab today. Okay, this is for animal cells or cells that do not have a cell wall. If you were to look at plant cells or cells with cell walls, then it changes a little. Hypertonic still has the same effect. Water is going to exit, but as water exits the cell, it causes the plasma membrane to tear away from the cell wall, and this is called plasmolysis. Plasmolysis. This was also written on the whiteboard today in lab. Okay, so the direction of the water movement is the same here, hypertonic, same for the plant cell as the animal cell. Water exits the cell, but the term, right, for plants is plasmolysis because you have this ripping of the plasma membrane away from the cell wall. Isotonic, though you have the same amount of water coming in and leaving the cell, under isotonic conditions. For a plant cell, this is not the preferred condition. And the reason for that that is Abby, my cat. Okay. So the reason that plants don't like isotonic conditions is because they actually prefer to have very full vacuoles that press the cell against the cell wall. And this is the turgid cell. It's hypotonic conditions is the preference. For red blood cells, a hypotonic condition would burst it open, but the plant has a rigid cell wall, and so it actually prefers the hypotonic. Okay, so make sure you can think back and forth between with and without a cell wall under these different tonicity examples, okay? And this is just like you saw with the celery today. If you don't water it, a plant, it will end up looking like you've put it in an isotonic situation, and it will wilt. Once you water it, it should perk back up. With the celery, instead of drying celery out, we actually put it in a hypertonic solution to cause the water to leave the cell. And then it was very, very wilted and limp and dying. For the isotonic, we soaked it in isotonic solution long enough to where it just became a little bit wilty, a little bit limp. So it's not exactly the same example, but similar. Plants typically can take on water and become turgid again, unless it's been dried out for too long and the cells actually die. Okay. For this, just know about the top part of this slide. So protists, freshwater protists like paramecia and amoeba, they have those special contractile vacuoles that they can pump water out of the cell very rapidly. So they don't have to have a cell wall to prevent lysis because they have pumps, contractile vacuoles or water pumps to squirt water out of the cell constantly. Your book gives examples of freshwater fish and saltwater fish, and even our brain helping to regulate solute concentration in the body. I'm not going to test you on those specific examples, but it's good to look at it and make sure you understand it to see if you're understanding the concept as a whole. Okay, active transport. Now you're talking about going against a concentration gradient. When you go against a concentration gradient, you're going from low concentration to high. So you're trying to put more solute in an area where you already have a whole bunch. And so this is going to cause a pump to forcibly put those molecules into the area of high concentration. So you're going to have to use ATP or energy to drive that pump to do this, okay? Force it against the concentration gradient. Sometimes it's not just a simple concentration gradient, but it's actually an electrochemical gradient, which is the total, taking into consider the total charge in that area of all like charge particles. So when we think about protons, for example, any positive ion is going to contribute to the overall electrochemical gradient. So if you had a whole bunch of positive ions and you wanted to move H plus, and you think, well, there's no H plus over here, I can easily move it down its concentration gradient. But if you're moving it, trying to move it into an area that's already positive, it will be very difficult because of the electrochemical gradient. We'll take another look at electrochemical gradients on the next slide, but you're taking into account both the charge and the specific substance. And either one can be enough to slow down or prevent that movement and then cause you to have to do an active transport. Furthermore, another layer to active transport is that it can be primary, where you see the ATP get used directly, as in this picture on the right, or it could be secondary, where it's relying on a primary transport that occurred somewhere else in the cell, but ATP is not being directly used in the secondary active transport, but it's only able to occur because of a primary active transport that already happened. Okay, so I know that this is the hardest slide of the lecture. Okay, so make sure you spend time understanding active transport, electrochemical gradients, and primary and secondary active transport. That's the hard stuff. Okay, so one more time with electrochemical gradients. See this picture, you have sodium chloride ions, negative and plus. You have a potassium ion, plus. You have these large grouping of negative charges. This could be like a protein in the cell, and proteins tend to have negative charges associated with them. So inside the cell, you have a lot of proteins, so you have a lot of negative charges, and they're going to attract positive charges to them. So you have these potassium positive charges attracted around these negative charges. And then you have a little bit of chloride and sodium, but not very much. And so this sodium really wants to come in. It's attracted to this negative charge. It's also wants to come in because there's a lot of it on the outside and almost none of it on the inside. But the cell is not permeable to sodium and it keeps it out. And if it was permeable, it would start to come in until either it reached equilibrium or there weren't enough negative charges to attract it anymore. So electrochemical gradients are the combined effect of both the concentration gradient of the substance itself and the total electrical gradient present. And there will be a difference between the inside of the cell and the outside of the cell. An electrical gradient, where the cytoplasm contains more negatively charged molecules than the extracellular fluid, is a major concept when thinking about the body and how the cells of the body function. And the classic example is thinking about your nervous system and how nerve impulses can be produced and transmitted. Okay, so continuing with active transport, this is using a pump to force a solute against the concentration gradient. You could have one particle being transported, and this is a simple uniporter for one molecule or ion. A symporter, on the other hand, you have two different molecules or ions, and they're going in the same direction. So taking two different substances and bringing it inside. Or you can have an antiporter where you have one substance coming in and another substance going out, so going in opposite directions. So classic example, all of the cells in the body have these pumps called sodium potassium pumps, and they specifically will take sodium, remove it from the inside of the cell, pump it out. And then it will at the same time take potassium and bring it in. And it does this in a 3:2 ratio. So 3 sodium pumped out, 2 potassium pumped in. And to do this pumping, it takes 1 ATP. So that's what this picture is showing here. Sodium and potassium. Okay, so you have sodium, 2 of them, and 3. 1, 2, and 3. 3 sodium will be pumped out, and here's released 1, 2, and 3. And then 2 potassium are taken in. And it is, I understand how it's confusing that it's only using 1 ATP because it looks like 2 separate processes. But when the 3 sodium bind to this protein, it causes this ATP to bind. And that binding of the ATP releases the sodium and allows for the potassium to bind. And then the ATP is hydrolyzed or split to release the energy to then force these 2 potassium into the cell. Okay. All right, so thinking about uniport, symport, and antiport, would the sodium potassium pump be uniporter, symporter, or antiporter? Next question, thinking about this pump, sodium potassium pump is called electrogenic. It's a type of electrogenic pump. What do you think that means? Okay, you can pause the video and answer these 2 questions. Okay, so this is a antiporter system or pump because you're taking in 2 different molecules and they're in 2 different directions. An electrogenic pump means that you're creating a charge imbalance, right? So you're putting more and more potassium inside the cell, where you're only bringing in 2 potassium at a time. You're releasing 3 sodium ions from the cell. So you're putting out more positive charge than you're bringing in. So you're putting more positive charge outside and contributing to keeping the inside more negative. Okay. So secondaries. So this is an example. The sodium potassium pump is a primary active transport because you're using ATP directly. Secondary active transport, now you've created a gradient with primary, you can use that gradient for secondary movement. So let's see. So you have a proton pump, it creates a gradient. Then you have this other protein that is going to capitalize on that gradient, allow a little bit of protons to come back in. That provides the energy to force sucrose against its concentration gradient. It's so energetically favorable for a couple protons to come back in to the cell that it allows sucrose to be pushed against the concentration gradient. Okay. So another example is looking at the sodium potassium pump that we just learned as the primary active transport, and then looking at a glucose pump where glucose goes down the concentration gradient. There's more glucose inside the cell, but you want every bit of glucose to go into the cell as possible. So you're going to go against the concentration gradient by allowing a little bit of sodium to come back in where there's almost none and there's a lot of sodium out here. So the sodium really, really wants in. And by letting a little bit in, you can get that glucose in against the concentration gradient. So secondary active transport It took energy to make this sodium gradient. Now you're able to use that gradient to drive the transport of glucose. So it's secondary active transport. Okay, so this is everything we talked about today. Review it, memorize it, make sure you understand it. Bulk transport, if it can't fit through the membrane and it can't fit through a protein, then you're going to have to use vesicles to transport in and out of the cell. And that's called bulk transport. This will take some energy in addition to the vesicle. Okay, so you can import, and it's called endocytosis, to bring it inside, or you can export to leave the cell, and that's called exocytosis. It's going to exit the cell. There are 3 types of endocytosis: bringing in materials, bringing in food, bringing in resources. It could be phagocytosis, pinocytosis, or receptor-mediated endocytosis. And then for export, it's just one example. Please make sure you're watching the video links, even though I'm not showing them. Okay, this one is different from this one. Watch them both. Okay, endocytosis, you get what we call invagination of the membrane. The membrane folds in to surround the particle of interest. It makes a vacuole, okay? And then the cell can decide what to do with that vacuole. So that's phagocytosis. We already saw this example when we looked at like an immune cell eating a microbe, and then a lysosome would fuse with the vacuole to break it down, kill it. But sometimes phagocytosis is performed to like eat food or resources. Okay, so your example here, I stuck with the neutrophils and macrophages of the immune system because that's what we saw in a previous chapter, killing a microbe pathogen. But here it's just a particle. It could be food, okay, like amoebas do. Pinocytosis is similar, but it's much smaller, and it's considered cellular drinking because you're taking in a small volume. That small volume could include a particle, or it could just be fluid. Okay, there could be dissolved, very small molecules, particles in here. Okay, so think of small. So pinocytosis, cellular drinking. You're still getting the folding in and pinching off of the membrane, but now it's surrounding a small bit of fluid. It's considered non-specialized, and this is smaller vesicles than you would see for phagocytosis. Would be bigger. Okay, receptor mediated. This is specific due to specific binding on a receptor. So a receptor is only going to bind to one specific substrate. If that specific substrate binds, it can start the process of folding in that membrane onto itself. And there's more receptors involved to deal with the next steps of dealing with what you caught, what you brought inside. So receptor-mediated endocytosis is the uptake of a specific substance using specific receptors. When that substance binds to those receptors on the external surface of the cell membrane, it brings that substance inside and traps it in a vesicle. This vesicle is coated with protein because it happened at a region. Those receptors are going to be on regions where there are other proteins on the inside. And this example, this protein on the inside is called clathrin, but it's not always that, but it is the most common example. So clathrin dependent Clathrin proteins are located on the inside part of the depression, which is the opposite side of the receptor. Okay, so this will be at cell depressions, and an example of what it could be bringing inside would be like LDL. LDL stands for low density lipoprotein. This is what we consider bad cholesterol. But there are lots of other examples of for receptor and substrate. There's also other times where the protein on the inside of the cell is something other than clathrin. Okay. And then lastly, and then we're done, exocytosis. Exocytosis is when the cell has stuff it wants to get rid of, and then it makes a vesicle, and that vesicle will then fuse with the membrane, and that will release the substances. So we do this with proteins. We saw when we looked at the Golgi apparatus, for example, proteins, hormones. When you think about neurotransmitter that's happening at synapses, this exact process, okay? And so then here is a summary of what we talked about in lecture today. In class, we were going to split into groups and answer some of the questions that I didn't assign for homework. So what I'm going to have you do is attempt to answer them by yourself. So you can see the question and then pause the video, type or write out your answer to see if you get it correct, and then you can check your answer. So go ahead and try to answer this first question. Why is it advantageous for the cell membrane to be fluid in nature? Okay, did you pause the video? Did you answer the question? Okay, so the fluid characteristic of the cell membrane allows greater flexibility to the cell than it would if the membrane was rigid. It also allows the motion of membrane components. Remember that mosaic of all those components embedded in the membrane? It allows things to move around. And sometimes this movement is also required for membrane transport, like the bulk transport we just saw. Okay, how can a cell use an extracellular peripheral protein as the receptor to transmit a signal into the cell? Pause the video and try to answer it. Okay, peripheral proteins can bind to other molecules in the extracellular space. However, they cannot directly transmit a signal to the inside of the cell since they do not have a transmembrane domain. So they must associate with integral membrane proteins in order to pass the signal to the inside of the cell. Did you get this one? This one was a little tricky. Okay. Next question, discuss why the following affect the rate of diffusion: size, temperature, and density. Oh, and distance. Okay, pause the video and try to answer. Okay. Heavy molecules move slower. Hotter temperatures speed up molecular movement. It's going to speed up diffusion. A denser solution is harder for molecules to move, slowing it down. Okay, so living cells require a steady supply of nutrients and a steady rate of waste removal. If the distance the substances travel is too far, diffusion won't be able to keep up with the demand for nutrients and waste removal, and it will not be able to survive. Glucose is digested from food, enters intestinal epithelial cells. It's doing this through active transport. Why do you think this body uses active transport instead of diffusion, when all the other cells in the body are going to use diffusion, facilitated diffusion. Pause it, think about it. Okay, intestinal cells are exposed to environments that fluctuate, right? You eat, you have a big amount of nutrients, but then between meals, you have very little. And so you need to be able to get the food when it's there, and you don't want it to, you don't want glucose to start going from your intestinal cells back into the gut. So if you were operating just on diffusion, when you don't eat, the glucose would diffuse out into the lumen of the gut. Our body cells don't have to worry about this because we maintain our blood sugar. This is why you have to maintain your blood sugar. So if you're not diabetic, like people who are not diabetic never have to really worry about this because the liver will regulate your blood sugar just fine. But if you're diabetic, you have to constantly check your blood sugar, not only to make sure it's not too high, but also to make sure that it doesn't get too low. And so it's a lot of work for diabetics. Our body monitors this and is supposed to regulate so that you always have enough glucose in the blood so that your tissues don't lose their glucose. Okay, so active transport proteins ensure that glucose moves into the intestinal cells and cannot go in reverse. It also will maximize the amount of energy that you're able to get from food. So when cells already have a lot of glucose in them, you can continue to add more and get all the glucose from your food. Okay, so our gut needs to use active transport. Okay, almost done. The sodium-calcium exchanger transports sodium into and calcium out of cardiac muscles. Describe why this transporter is classified as a secondary active transport. Okay, so the sodium-calcium exchanger moves sodium down its electrochemical gradient. It wouldn't have an electrochemical gradient to use if you didn't already create it with a sodium-potassium pump. So the sodium-potassium pump is the primary active transport using ATP to create this electrochemical gradient. And then the calcium transport is able to utilize this gradient that you've already established. So it's secondary active transport. Okay, so your homework is to answer the review questions 40 through 20 and the selected critical thinking questions 22, 27, 28, 30, 33, and 34. All right, that's it.